with clinical assessment, diagnosis, and research in psychopathology. So we want to find out what assessments and diagnosis really means, what are those basic components and methods of research in psychopath, and then what role do genetics, culture play, what are some ethical principles to consider in research. So when we set out to do assessment, whenever a client walks in the door, let's put it this way, when a client walks in the door, you have to determine why they're there. Now, they will be able to self-report some information, of course, that's valuable, but you'll also need to be conducting an assessment process in order to determine several things. 1, to understand the individual. 2, to predict the behavior. 3, to plan the treatment. And then 4, to evaluate the treatment outcome. So they'll come in, they'll present with some issues, but then you're going to be thinking very comprehensively and saying, okay, I'm going to need to conduct this assessment to get to know that person, predict, plan, and evaluate for them. When you kind of think about assessment, it's like a funnel. It starts out very large, through a multidimensional approach, and you go from very narrow to very specific problem areas. One of the things when we're selecting our assessments, we want them to be both reliable and valid. Reliability, remember, is consistency in measurement. It includes all different types of reliability. How would you know that a test is reliable and valid? You typically get an administration manual with this test that you have bought. You're part of your overhead as a practitioner, and inside that administration manual is going to have all that different research. It's going to show how they've determined the reliability and validity of that particular assessment instrument. And it could be something like test retest, it could be inter rater reliability, and then of course it's going to discuss several different types of validity. And what you're wanting to see is whether or not the assessment tool actually measures what it's supposed to measure. How well does it measure? And that can include a discussion about concurrent, discriminant, and predictive validity. We want to see if the instrument is standardized. In other words, is it consistent in how you're supposed to administer it? Is it consistent in terms of not just administering it, but scoring it? Is it standardized in how you score it? You use populations for benchmark comparisons. You may have a structured administration that you follow. You may have structured scoring and a very structured evaluation procedure that it has to be followed. Which that's all very good because that lends itself to that question of validity and reliability again. So again, the assessment needs to be reliable. In other words, it measures something consistently. It needs to be valid. It's to the degree to which it really does measure what it says it measures. And then it needs to be standardized in terms of how it's administered, how it's assessed, etc. Types of assessments are going to include clinical interviews, and this is the most common. Clinical assessment can be either structured or semi-structured. In other words, a structured is going to follow symptomology that may be represented in the DSM-5. A semi-structured is going to have some of those questions that are very much based in the diagnostic criteria, but then you may add some additional pieces of questions that you want to get to some, maybe a little bit more depth observations that you've made, therefore that lead to questions, etc. You're going to do a mental status exam, which is, do they know what's their appearance, their behavior, what's their thought process, are they aware of time, place, and person, what's their mood, what's their intellectual functioning, what are the sensations that they're experiencing. And then a physical exam, you can make a referral to a physician there should you think that there's something other physically going on that could contribute to or a medical condition that could contribute to psychopathology there. All right, so you can see examples of the components of a mental status exam, and you can see the outcome for Frank on the right there. There's also behavioral assessments, which focus on the here and now. There's direct observations of the behavior in the environment, and the purpose of these tests is to identify problematic behaviors and situations. And as you're doing a behavioral assessment, what you're trying to do is you're trying to assess the ABCs, the antecedents, the behaviors, and the consequences that that person was experiencing. So that through observation. And that can be both formal and informal. It can be self-monitoring, in other words, by the client, or it can be being observed by others. However, the problem with the reliability of that is that sometimes people that are being observed perform. And so it can either fake good or fake bad with regards to psychopathology. So psychological testing should be both reliable and valid. There are different types of tests. There are objective test and projective test. Most of us are aware of the ink blot test. It's been around forever. I read a really interesting book this summer on Rorschach and what the original Rorschach actually intended for his assessment tool to become. It was not really an assessment tool at all. It was simply a measurement of what a person experiences. So once it got into the hands of everybody else, though, it became a tool that was rooted in psychoanalytic tradition. It was used to assess the unconscious process. It was used as a test to determine pathology. What happens there is that the personality, according to Freud, the id, ego, and superego, that those instincts get projected up out of the unconscious onto a neutral stimulus, and thereby then reflect what are the issues that those instincts are driving us towards. What is the abnormality that those instincts are driving us towards? A lot of inference that goes on in the scoring and interpretation, and they've tried throughout the years to make it a much more objective, valid type of scoring system in which they've not really achieved consistency even after all these years. So examples of those projective tests are the Rorschach, as I've said, the thematic apperception test, and the reliability and validity of both those things have a tendency to be very questionable. Here's an example of a Rorschach ink blot. See what you see there. And so, again, many people see different things, but according to Freud's theory, you're going to project your sexual and aggressive kind of instincts onto that neutral stimulus. I mentioned objective tests. There's also personality tests. The objective tests have very much an empirical tradition. They test the stimulus a little less, that are less ambiguous. They require a little less clinical inference and scoring and interpretation, which that's good because that's then going to remove the bias. And then there's personality tests like the MMPI, Minnesota Multiphasic Personality Inventory. This particular test has been around for quite some time, has been researched to death, and has been proven to be very reliable, valid, and it has a very normative base that has wide applicability. There are intelligence tests, and what we're looking at is we're looking at the nature of intellectual functioning and IQ, originally established to measure children's performance. There's a deviation IQ that allows us to compare to other people's IQs, and there are both verbal and performance domains. Now, in addition to that, there are some IQ tests that are changing, and so where they're not so much verbal and performance domain based as they are based in information processing. And these tests have been changed like that in order to reduce the bias there, because not all of us have that same exposure to verbal and mathematics type of processing opportunities. Neuropsychological test, purpose and goals is to assess broad range of skills and abilities. Goal is to understand that brain behavior pattern. Lurie and Nebraska and the Halstead-Raitan batteries are those two typical neuropsych tests. You have to have additional training as a clinical psychologist to become maybe a neuropsych assessor, tester, examiner is the word I'm looking for. All right, so the test really does range from grip strength to sound recognition, attention, and concentration. All right, so the problem with neuropsych test is that they yield false positives. In other words, show that there's a problem when maybe there really is no problem. Or they yield false negatives, fail to detect a problem that really is present. So those are kind of paper and pencil or computer-based tests, and practice kind of hands-on kind of tests. But with the development of neuroscience technology, we're really moving. I think eventually we'll move towards much more higher validity type of neuropsych-based testing. All right, there's also neuroimaging of the brain, and which is there to understand the structure and the function. There are CAT scans, PET scans, MRIs, functional MRIs. All of those things are looking at the electrical functioning of the brain. EEGs look at the electrical functioning of the brain. CAT scans are utilizing X-rays to look at the brain. Magnetic resonance imaging is utilizing magnetic fields, better resolution than CAT scans. Functional MRIs are looking at oxygen flow in the brain. PET scans are positron emission tomography, and they look at heat in the brain, and can involve, another example might be single photon emission computed tomography or SPECT studies, and both of those were interjecting radioactive isotopes into the bloodstream to measure all the glucose, the oxygen, and the blood flow in the brain. So lots and lots of big time technology that has developed across time to really, for us to be able to break down the structures of the brain and the functioning of those neurotransmitters. And so, which really it allows us to centrally locate where maybe some causal factors or indicators of psychopathology are originating from and how it's different from normal brains. All right, so psychophysiological assessment is where we're looking at this function, structure, and activity of the nervous system. And there's the psychophysiological assessment domains. EEG, I said, was the electrical functioning of your brain. There are tests where we look at heart rate and respiration. Then we can look at electrodermal response levels, which are sweat glands. And so, all of those things are important psychophysiological assessments. But then again, we also have to question the validity and reliability and how that information is actually going to be used. Lots of pathologies that have a very, very strong physiological component, such as PTSD, sexual dysfunction, sleep disorders, hypertension. So those assessment tools become very valuable in those circumstances. Classification, we talked a little bit about, I think in earlier sections where we referenced Kraepelin and his development of the classification system and how the diagnostic classification system is kind of a representation of the biological tradition. But classification is central to all sciences, and assignments to categories of symptoms are based on shared attributes or reactions. So it's not just willy-nilly. There are two approaches to looking at classifying symptomology. There's an idiographic as well as a nomothetic. An idiographic is we're asking what's really unique about that individual, their personality, their culture, their circumstances, whereas nomothetic is that we're trying to really use maybe a larger group, larger group, when we're trying to really arrive at a very specific psychological disorder. In other words, to make a particular diagnosis. So idiographic, really kind of looking and seeing what's unique about that individual. Nomothetic is when we're really trying to nail it down to, the psychological disorder or the diagnosis overall. So in classification systems, there are different types or terminologies representing those classification systems: taxonomies, nosology, nomenclature. So a taxonomy is the classification in a scientific context. Nosology is the taxonomy in a psychological medical phenomena, and nomenclature is a label in nosological systems. So in other words, taxonomy would be like, what are the scientific principles and or structures of this particular set of symptoms. Nosology would be what is the medical diagnosis, the psychological diagnosis, and what are the symptoms that represent that disease. And the nomenclature is simply what's the label that you're going to put on that category. So there are categorical and dimensional approaches. The current DSM system is that thing. It is both a categorical and dimensional approach. Now, the pure categorical approach is what we used to do. And it was very much you either had the symptoms and therefore had the disease or the disorder that you didn't. Dimensional approach is you're classified along dimensions. In other words, people have differing amounts of that particular disorder. And prototypical approach that we're trying to accomplish now in psychology is a combination of both those things. So the Diagnostic and Statistics Manual of Mental Disorders has been updated every 10 to 20 years. The current release is DSM-5 that was released in May of 2013. Previous edition was the DSM-4TR. Now, that's what you use in classification if you're seeing a psychologist. That's their Bible. That's their classification system. That system is actually based upon the ICD system, which is the International Classification of Diseases produced by the World Health Organization. And that's where the disease model has kind of influenced the diagnostic manual for psychology in that they're similar. You have codes, that you check off whenever there's a list of symptoms that meet that particular code, and that code then is understood by everyone in the field as representing that particular syndrome's symptoms, behaviors, emotions, presentations, frequency, duration, time periods, etc. So prior to 1980, diagnosis is based on either biology or psychoanalytic theory. With the DSM-3 in the 1980, classification is revolutionized. In other words, we're trying to increase our ability and reliability in terms of classifying and talking about those symptoms lists. Diagnosis at that time is made upon 5 axes. And what does that mean? In those axes, it's describing the characteristics of the problem and the source or the causality of the problem, etc. So, for example, in axis 1, you'd be saying, okay, well, axis 1 is the reasons why the person is coming for clinical care. Axis 2 would be if that person had a personality disorder or mental retardation. Axis 3 would be those medical conditions that we think could possibly contribute to the onset of the psychological disorder. Axis 4 would be those psychosocial factors, family, environment, school, neighborhood, those things that we think could impact the onset and the presentation of that psychopathology. And then axis 5 is what we call a GAF, a global assessment of functioning. And on that particular axis, we're saying, okay, now how well is this person functioning? Do they need to be out and about? Do they need to be hospitalized? Are they in crisis? What kind of care and treatment level do they need to be involved in? So DSM-4 comes in around in 1994. It eliminates the difference between psychological and organic mental disorders. It says essentially that there's a much more integrative approach, that illness is actually due to psychopathology is due to both psychological and biological factors. And then DSM-4 comes along, and it just is simply an update as to the new research, some slightly altered criteria for certain diagnostic categories. So what did DSM-5 do? It removed the axial system. It gave very clear inclusion-exclusion criteria for disorders. Their categories disorders are categorized under very broad headings, and it's very much empirically prototypic approach to classification. There are problems that are still unresolved in DSM-5. The issue of comorbidity, where 2 or more disorders are occurring for the same person. We're knowing that high comorbidity is actually pretty common, and it emphasizes reliability, maybe at the expense of validity. In other words, we may kind of superficially and artificially split some diseases that really don't need to be split, but they're very similar. The dimensional quality to DSM-5, though, was intended to move towards a more dimensional approach. It says but most critics don't think that our dimensional approach is better than what the DSM-4 was doing. And we still have the issue with labels and stigmatization. So what do we want to do with assessment and diagnosis? We want to understand clients. We want to understand and ameliorate suffering. We want it to be reliable, valid, and standardized. When we conduct research, we can classify all day long. We can sit around and diagnose all day long, but we need to know whether or not that symptomology really, truly, with validity and reliability, reflects that particular diagnostic category. Okay, so how do we set about doing that? Well, one of the things we have to understand psychopathology. So we want to know what problems cause stress, impair functioning, what causes people to behave in unusual ways, and what causes some people to behave in more adaptive ways. So we start with a hypothesis. However, not all hypotheses are testable, but we want to make them into a testable format. We have to select our research design, which is our method for testing that hypothesis. We have to define an independent variable, which is that variable we're manipulating or that influences behavior, the dependent variable, which is the outcome variable that we want to look at. In our research design, we want to really consider validity and reliability. We want to have good internal as well as external validity. The internal validity is going to help us to say that, yep, these results are really due to the independent variable. External validity are going to allow us to generalize it beyond our sample to the population. So how do we increase internalizing? How do we increase internal validity? Reducing confounding variables. And how do we reduce confounding variables? Those variables that may mistakenly cause us to accept another outcome. So we control those confounding variables through control groups, random assignment procedures, and analog methods. We have to decide on our statistics that we're going to use. Are we doing descriptive? Are we doing inferential? Combination of those things. So what is stats? It's a branch of mathematics that helps us protect against bias. We're looking for statistical significance. In other words, are these differences due to chance? We're looking for clinical significance. Does it seem to have clinical meaning for my client? And it doesn't. Statistical significance doesn't always mean clinical significance. So how do we evaluate those two? We look at the effect size coming out of our data. We look at the social validity. All patients are not the same. Some researchers have a tendency to think of all patients being the same. That affects how information is collected and interpreted and used. Generalizability is that whenever we're taking our information and we're saying these results are applicable to the larger population that has been studied. When we study individual cases, we can do case method study, which is really very descriptive about one client. It involves observation. That's what many historical developments in psychology were based upon, not necessarily the most valid and reliable approach. There are definitely those limitations. Again, validity, reliability, lack of scientific rigor, lots of confounding variables. We can then do a correlational study, which looks at the relationship between the variables. The nature of correlation is that it's going to be the statistical relationship between two variables. There's really no manipulation of an independent variable. Your correlation can range from -1 to a +1 to a negative to a positive correlation. So now why would we use correlation? Because we can't manipulate the independent variable. There are definitely limitations to it in terms of we can't talk about cause and effect, can't talk about the direction of the outcome or the relationship. But we can do one form of correlational method called epidemiological research. And this is important research that the CDC, Center for Disease Control, does. They look at incidence, prevalence, and course of disorders, such as AIDS, such as trauma. And we know that we can have both positive and negative correlations as well as no correlation. In other words, if you look at this relationship between age and sleep problems, looks like, you can see that as people grow older, for the positive correlation, as people grow older, they tend to have more sleep problems. Then for the negative correlation, as people grow older, they tend to have fewer sleep problems. So we kind of go, okay, well, wait a minute, which one's true? And then there's this thing about, in this third study, where we have no correlation, it appears to be there's no relationship between sleep problems and age. So that's correlation in case study. Now we're going to pop over to experimental method. And this is where we do manipulate the independent variable. And we're looking for cause and effect. And how do we do this? We do this by assigning people to either control groups or to experimental or treatment groups. In a control group, it can be either you're getting a placebo or you can be in a double-blind type of control group. Placebo is you're given a sugar pill. You don't know that you're not really getting the treatment. Double-blind is that the participants and assessors are both unaware of what kind of treatment the participant's getting. All right. We can do comparative treatment designs. In other words, type of group design where we can compare treatment in similar persons and then look what the process and the outcome has been like. Single subject designs do have strengths. It's a rigorous study of single cases. It varies experimental conditions, time, there are repeated measures. There's a evaluation of variability level and trend, and it has a pretty good internal validity. So what are they? Withdrawal designs, multiple baseline designs. This area in the field of behavioral genetics is a really hot area right now in psychology, looking at the interaction between genes, experience, and behavior. Genotype, genetic makeup, phenotype versus genotype studies where we're looking at genetic makeup versus observable characteristics. And some of those genetic studies involve families, adoptees, twins, looking for those genetic links and association studies there. What's the rationale and overview for doing research? How does the behavior present? How does it look? How does it change? How can we prevent it? And how can we treat it? We can study that kind of abnormality over time through cross-sectional, longitudinal, and sequential designs. And there are definitely assets and liabilities to both of those. We can look at cross-cultural research. In other words, overcoming ethnocentric kind of research, where we're broadening out across multiple cultures and looking at the differences there and the context, considering the context there. So what the assets there would be that we're able to see how psychopathology manifests itself in other ethnic groups. But the problems with cross-cultural research is that it's limited by its measures. So what are the programs of research? What are the components? Set of interrelated research questions drawn on several methodologies. It's conducted in stage, going for a replication. And replication is critical so that we can ensure that we're protecting against fluke results. Of course, everybody has to go through the IRB, which is and follow the APA code. IRB oversees the rights of human subjects participating in research. And APA's ethics code makes sure that you're handling it correctly, in addition to what the IRB does. So the IRB enforces a set of ethical principles that have to be proven with every study: informed consent, competence, voluntarism, full information comprehension. So informed consent, that developed post-World War II in the Nazi era. Competence is that you have to be able to provide consent. Voluntarism is that you're not coerced. Full information means that you have all the information you need to make your decision to consent. And comprehension means that you understand what the risks and the benefits of their participation is. So research is set out to do establish and testing of hypothesis, and research designs depend upon the questions that's being asked and the benefits and the drawbacks. Abnormal psych, the study of abnormal psych is founded in the scientific method. We're looking at nature, causes, and course, and then prevention and treatment. Replication is critical, as has been said, and